Alrighty, good morning. Um, we're going to go into the process of photosynthesis, a little bit more detailed than I think you've done before. But we're going to start with the definition of what photosynthesis is. It's just the process of turning light energy from the sun into chemical energy, specifically into sugar or glucose. So I didn't put it on the arrow here. I should have put it in the text box, but usually above this arrow right here is the word light energy. So you understand that it's not really a reactant, but it is necessary to get the splitting of the carbon dioxide in the water to create the sugar, oxygen, and water. So I've got a balanced equation here, and so this is the equation for photosynthesis. I can't remember if we went over cellular respiration or not, but this is the opposite reaction of cellular respiration, so they're coupled reactions. So the reactants of this photosynthesis then become the products of photosynthesis then become the reactants for cellular respiration, and their products become photosynthesis reactants. So it's a nice big cycle there. So light energy, thermal energy, captured, turned into that chemical energy of the sugar. So we're going to talk first about the absorption of light. We talked about this when we were going over the biology parts of the cell. The pigments within the plastids are able to absorb different wavelengths of light energy. They don't absorb all of them. So if they absorb some, they're going to reflect others. In the case of chlorophyll, which is the main pigment used in photosynthesis, it's going to absorb red, blue, and violet wavelengths. And then it's going to reflect the green and yellow. So this is why your plants appear green, because they're reflecting that green and yellow pigment or wavelength. So if you check out your first diagram that I've drawn you, it's the visible light spectrum there. From the left to the right, you lose energy. You also gain a longer wavelength. So short wavelengths are going to have more energy, longer wavelengths will have less energy. And everything's, like I said, like I've noted there, is measured in nanometers. So what ended up, I guess I'm ahead of myself, sorry. Here we go. What, I'm going to go to the, I'll just start here. With the action spectrum, this is an experiment. Well, it should be, sorry, I'm reading my 3 different pages here. Your action spectrum, this is an experiment that tests the effectiveness of different wavelengths of light to do certain processes. So what wavelengths are required for certain chemical reactions to occur. So Engelmann did an experiment back in 1882, 1883, depending on the source, where he used some green algae and put them on a slide, and he shone a light onto that slide. He projected a light onto the slide, but before the light hit the slide, it went through a prism in order to separate the different wavelengths. So the diagram's kind of showing you that we've got the red on the left and the violet on the right. And so he was he was going to try to see what wavelengths were necessary for photosynthesis. Since the green algae are oxygen dependent, they're going to be able to photosynthesize and they're going to need oxygen. So he wanted to know which wavelengths were necessary for photosynthesis. And so what he found out was that the green algae tended to collect around the red light waves and then the blue and violet. So that implies that they needed the red and the blue and the violet wavelengths to go through photosynthesis. And they didn't necessarily need the green and the yellow colors to do that, to go through photosynthesis. So, all right. There's a couple of photosynthetic pigments that I want to draw your attention to. There's chlorophyll pigments and carotenoid pigments. So with the chlorophyll pigments, there's two different types. There's chlorophyll A and chlorophyll B. They're just different versions. And then you've got your carotenoid pigments. You've got carotene and xanthophyll. So, I'm gonna give you a second to write all this down, I think, first, and then I'm going to explain the diagrams of the process of this first part of photosynthesis. So the very first part of photosynthesis is characterized as the light-dependent reactions. Tomorrow we'll talk about the light-independent reactions, or the dark reactions. So obviously from the words light-dependent, it means that it requires light in order to occur. So without light, you're not going to go through these two processes. It's going to occur in the thylakoid membranes of your chloroplasts. And these light-dependent reactions are entitled photosystem 1 and photosystem 2. The reason why they're out of order, because that's kind of one of the first things that I noticed when I learned about this, is you have photosystem 2 before photosystem 1. Photosystem 1 was discovered first, and then photosystem 2 was discovered second. So, but they figured out that photosystem 2 actually came before photosystem 1, so rather than renaming it, they just kept it as photosystem 2, and then you go through photosystem 1. Photosystem 2 also includes the photolysis, or the splitting of light. Your photosystem 1 is going to include that cyclic electron flow down at the bottom. So, and then in between photosystem 2 and 1, we're going to go through the non-cyclic electron flow and the non-cyclic photophosphorylation. Sorry, these are really long words. So interesting to say there. So, and by the way, there is no reason why on the Z scheme down here, non-cyclic and non-cyclic, they don't have a dash there. They should. So, but like I said, I'll explain what the non-cyclic electron flow and the non-cyclic photophosphorylation and the cyclic photophosphorylation are when we get into the pictures, because I think they'll make more sense that way. Okay. The last thing that I want you to know, besides the fact that we've got the different chlorophyll molecules, is I want you to know that we also have these things called antenna molecule. So, which can be your chlorophyll A and B's. So, okay. Go ahead and look at your diagrams. So your very first diagram of the process of photosynthesis, we have I've got the sunlight going through the top portion of the leaf into the mesophyll. It doesn't really matter if it's the spongy or palisade at this point, and then I've got the chloroplast there. Remember, the chloroplast is a double membraned molecule, and so that's what you're kind of looking at right now in the next picture over. So, okay, so the large molecule there kind of in the center, that's a chlorophyll A. And it's called the center molecule. This is where the main reactions are going to occur. Your antenna molecule are coming off of that chlorophyll A, P680. And so, but they're just different types of chlorophyll A or chlorophyll B molecules. Okay, but collectively they're known as antenna molecules because they're not actually going to do the actual chemical reaction. Their main purpose is just to pass along the light energy. That's it. Because the sunlight isn't always going to hit directly onto that chlorophyll A, P680. Sometimes that's why they have those antenna molecules spread out. So that way, if it hits one of those, those will then pass the light energy on until they get to that chlorophyll A, P680. So this is known when the antenna molecules capture that light energy and then pass it on to that chlorophyll A, P680. That's what's known as resonance transfer. So resonance transfer is just energy from one antenna molecule gets passed to another and then on until it gets to the chlorophyll A, P680. So that's the overall process. That's going to happen in both photosystem 1 and photosystem 2. Is you're going to get that resonance transfer as the light hits those antenna molecule and passes it on to your chlorophyll A molecule, your center molecule. So specifically, the next picture, what happens in photosystem 2? So you've got your light energy coming down, and it's popping over. Once it gets to that chlorophyll A, you're going to go through what's called photolysis. You're going to split your water using the light. Excuse me. So you're going to split apart that water into the oxygen molecule and then 2 hydrogen ions. And then the electrons from that are going to be passed up into a primary electron acceptor. Okay, so they're going to a high energy level at that point, up at that electron acceptor. I'm sorry, I don't know what's in my throat. Then what's going to happen are these electrons are going to fall. Oh, by the way, that is photosystem 2. Okay, and the important thing to note is that when you split the water there, that oxygen that's given off as the product, that is the actual oxygen that we will breathe in. That's the oxygen that the plants are giving off that we require. Okay, so with your photosystem 2, you've shot your electrons up into that primary electron acceptor. Now they're going to be passed through these electron carriers. Okay. As they fall back down into the next picture, you can kind of see them falling back down. What's going to happen is the adenosine diphosphate and an extra phosphate are going to be combined. So as the electron's coming down, it's going to decrease in the amount of energy. As it's going down, it's going to provide that energy necessary to bind ADP with another phosphate to make ATP. Okay. And so that ATP is then going to go into the Calvin cycle, which will be part of the light independent reactions, which we'll cover tomorrow. So that ATP that we're making between photosystem 2 and photosystem 1 will be used tomorrow. So as it comes back down, it's going to potentially hit antenna molecule, or it's going to hit a chlorophyll A molecule, but instead it's going to absorb a different amount because this one's called chlorophyll A P700. So the process of shooting those 2 electrons from the water molecule up to that primary electron acceptor and then allowing it to pass through those electron carriers down to the chlorophyll A P700 molecule, that is known as non-cyclic electron flow. It's not a cycle. It's a Z scheme. So specifically the part about splitting the water, it's non-cyclic. So that's why that portion just in photosystem 2 of splitting, taking the light energy and splitting apart that water molecule and shooting those electrons up, those electrons don't come back down to that chlorophyll A P680. They go across the way to chlorophyll A P700. That is called non-cyclic photophosphorylation. So, or I'm sorry, I'm so sorry, that's backwards. That's backwards. So the non-cyclic electron flow is the shooting of the electrons up to the primary electron acceptor. It's not going to come back down. The electrons going down through the electron carriers from 680 to 700, that's the photophosphorylation. I'm sorry, I got those backwards. Okay, so now we're in photosystem 2, or 1, sorry, photosystem 1. It's the second one, but it's called 1. So the energy that was captured in photosystem 2 that got excited by those electrons from the photolysis have now been passed on and captured by the chlorophyll A P700 acceptor. It's going to take that in addition to sunlight, which are going to hit those residual antenna molecule and pass that along to that chlorophyll A P700. And then what's going to happen is it's going to do kind of the same thing. It's going to shoot those electrons up, shoot those electrons up into another primary electron acceptor. In that process, we're going to take a coenzyme called NADP, and we're going to be able to connect another hydrogen with it to make NADPH. That NADPH is then going to be sent out, and we're going to use that in the Calvin cycle as well. Again, just like with photosystem 2, the electrons aren't going to just hang out with the primary electron acceptor. They're going to fall back down, but instead of going to a different molecule, they're going to cycle back down into the chlorophyll A P700. When they do that, as they're falling back down, as they're falling back down to a lower energy level, we're going to be able to make ATP there as well. And again, that ATP is going to be used in the Calvin cycle also. So That is basically the first two steps of photosynthesis, photosystem 2 and photosystem 1. So I've given you some other numbered pictures there. 1A and 1B, you've got your light. 1A, you've got your light hitting your pigment molecule, and you can do one of two things. It can give off heat or it can give off light. 1A is when it gives off heat, okay, as it falls back down. 1B is when the light hits the pigment molecule, gets shot up to that primary electron acceptor, and it comes back down as light. That would be like fluorescence. Now the thing to remember is if something is fluorescent, it's emitting light that's always at a lower wavelength because it's going to have less energy than when it was absorbed at. So then you go on to step 2, and you're going to pass that energy to a different pigment molecule, or assuming that you are an antenna molecule. So you're going to basically go through the process of resonance transfer at that point in step 2. So now the other thing that can happen with the third option there is light will come down to that pigment molecule, go up, and then that electron, the primary electron acceptor will just capture that energy. So it'll hold on to the energy that way. So I guess I should say they're not really steps, but 3 different solutions or 3 different possibilities of how light can be captured and then used in these first 2 steps of photosynthesis. So hopefully this makes some sense. If it doesn't, please let me know. Tomorrow we'll go through the light-dependent reactions, and I haven't finished outlining it. So I don't know if we're going to get through that in one go through, if it's going to require another one. So if you do have any questions, please don't hesitate to ask. And have a good day.